In this protocol, we describe a simple method to fabricate a paper-based device called the ExoPad, which is designed for the effective enrichment and isolation of extracellular vesicles. The ExoPad is a simple device that can preconcentrate and isolate extracellular vesicles without the use of any chemicals or bulky machines, while showing higher recovery rates than conventional methods.
Begin by defining the region to be printed with printer software. The design should have 12 wax-patterned layers in which the diameters of the circular sample areas gradually narrow from 5 to 2 millimeters. Then, print hydrophobic wax on the designated regions on both sides of the cellulose paper. Place the wax-printed paper in a laboratory oven for 80 seconds at 120 degrees Celsius. Then cut the wax-printed paper with a cutter to make individual ExoPad devices. Drop 5 and 2 microliters of perm-selective membrane onto the sample areas in the leftmost and rightmost layers, respectively. Place the layers coated with the perm-selective membrane on a hot plate at 70 degrees Celsius for 30 minutes to evaporate the solvent. Then, seal the outermost surface of the coated layer facing the buffer solution with pressure-sensitive tape, leaving a small hole. Fold the ExoPad device back and forth along the white lines to convergently connect all sample areas. Pipette 15 microliters of the microvesicle and exosome sample in the convergent sample areas.